From Microbe TV, this is Matters Microbial, a podcast about microbiology, microbiologists, and microbial centrism. This episode was recorded on January 3rd, 2024. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 23rd episode of Matters Microbial. As always, I continue to be grateful for every listener, viewer, and commenter. Please let me know if I can make any changes to improve the podcast. For example, last week, I called Indiana University the University of Indiana. Mea culpa, Hoosiers. I'll try to do better. First, a member of my Wunderkammer. As I've stated before, I believe that encouraging students to approach issues in science with creativity improves concept retention, ownership, and overall enthusiasm. It is by no means superficial. The late Lynn Margulis had a wonderful quote that's often with me. Beneath our superficial differences, we are all of us walking communities of bacteria. The world shimmers, a pointless landscape made of tiny living beings. You can imagine the effect that had on young Doc Martin. But as seen here, a micronaut from one of my former Bio 350 microbiology courses created this artwork to celebrate that quote. The image is framed and hangs both in my laboratory and in my office. That's right, I have two of them framed. I will include links about the complex and much missed Lynn Margulis in the show notes. It is amazing that it's 2024, friends. This coming semester, I'll be teaching Biology 111A, the unity of life, to almost 50 students. This introductory course covers the basics of cell and molecular biology, and the overall Bio 111 course logo, seen here, is by Katie McKissick, better known as Beatrice the Biologist, and every student gets such a sticker. Because this course is large, at a small undergraduate institution at my own, let me be clear, laboratory becomes vital. There's much more opportunity for professor-student interaction, which is crucial. So I use a cure-based approach based on the wonderful Tiny Earth program, logo is seen here, developed by Joe Handelsman, and I'll include links to that program in the show notes. The first thing that I do to teach basic microbiological skills is I have students streak out from their reusable water bottles, and they can discover the nature of their water bottle buddies, as seen in this sticker by Ethan Kokic. Then, we focus on trying to find, as Tiny Earth does, new antibiotic-producing microbes from soil, lichens, and mosses. Normally this is done with soil, but I'm interested in lichens and mosses for a variety of reasons. Students will look for interactions such as this one seen here. And here is the wonderful sticker created by the inimitable Liza Van de Art. I'll keep you posted about the adventures of Team Molecular, as I call them. One of the tragedies of our undergraduate educational system, I think, is how limited the introduction is to the amazing microbial world. There's so much to tell students about. The depth and breadth is immense. In particular, not much time is spent on fungi, other than yeast for beer and wine. Now, to be clear, the yeast system is a fabulous one for molecular biology and genetics, make no mistake. But the fungal world is amazingly diverse and understudied. I actually did some work in graduate school with Dave Perkins on Neurospora crassa, and got to study the fact that Neurospora can tell what time it is. With this idea, We are very fortunate indeed to welcome Dr. Arturo Casadevall of Johns Hopkins University to discuss his path in science, his thoughts on the different faces of the fungal world, and the remarkable breadth of his interest. No kidding that. Welcome to our Quality Quorum, Arturo. It's such a privilege to have you here.
Mark, thank you so much for inviting me. I'm happy to be here.
I think a good place to start, if you wouldn't mind, is to tell the listeners and viewers a little bit about your path to science, which I know is remarkable.
The path to science. Well, you can tell that I have an accent that places me having been born outside the United States. So I came to the United States at the age of 11, and I had your typical immigrant experience, settled in Queens, New York. We went through the usual difficulties that happen. We were not, I didn't come from a science family. So the only place I could go to school was the city university, which at that time was free. And I went to Queens College. And when I went to Queens College, I discovered science. And I remember being shocked that people got paid to do research. I didn't know where research was done. And once I found that you could make a living being a researcher, I was so interested in a career in science.
I know it's been, you have, you went to medical school.
Well, that was another part of the story. And if you want, I'll tell you about it. What happened was that we kind of knew difficult times when we first settled, and I always wanted to figure out how to get out of the situation I was. And somewhere on the line, I figured that if I could get really good grades, I may be able to find a way out. And I got good grades, and I decided that I was going to go I got accepted to a PhD program in chemistry. And when I told my dad about that, he said to me, I don't know about that. So he went and talked to somebody else who advised him that your son needs to go to medical school. And my father told me, we've been through a lot. I want you to go to medical school and you get yourself a degree and come back and practice in the community. And I said, Dad, I want a career as a scientist. And he said to me, you're going to medical school. And I said, okay, I'm going to medical school. I didn't have any of the pre-meds. I didn't have anything done, but, kind of figure out how to do it. And I did get into it. But in the meantime, I discovered that there was something called an MD-PhD program. And MD-PhD programs created the opportunity to be okay with my dad and also to pursue a career in science. And I was lucky enough to be accepted into one. And I was very glad that my father suggested that I study medicine, because being a physician has given me a lot of views into problems and into things that need to be investigated. But doing an MD PhD allowed me to combine the two, and I'm very grateful to have been accepted in that program.
I think it's a wonderful story. I'm glad you told it in that way. I'm first generation, and my parents would first off, to be honest with you, had I had wonderful grades, I would have been encouraged to go to medical school, and that wouldn't have set very well in this noggin of mine. So I'm actually very pleased with the route that I took, bumpy though it's been at times. Still, because I've always loved research, I've always loved science, and my parents never knew where that was from. Was it that way for you? I ask because you have so many interests.
Yeah, I have a lot of interests. I will give you one more anecdote from that. When I applied, they told me that nobody gets into MD PhD programs from this school, so you're wasting your time. And they were almost right, because I didn't get accepted until a couple of days before school started. You know what happened? Somebody dropped out, and they offered me the place. But I'm just very glad that they accepted. And when I told my dad that I've been accepted to this program and that the program came with a stipend and that they pay your tuition, he looked at me and he said, Arturo, what kind of racket is this? You mean to tell me that they're going to pay you to go study and they're paying you tuition? I said, Dad, there is some sort of grant that is basically to train physicians to do research. And he looked at me with suspicion, but it worked out.
I have to tell you how much this resonates with me. My father was a firefighter, and before that, a truck driver, and before that, an oil field worker. So he knew none of this kind of business. And when I had training grant support to go to graduate school, he refused to believe it. To the point when he dropped me off in Palo Alto, he said to me, his last words before he drove off, he says, "You be sure to call me when this falls through." And he didn't mean that I wasn't smart. He didn't mean there wasn't a program, but he refused to believe that they would pay. Now, to be fair, they didn't pay a lot.
I know. But
still, it's remarkable.
Still the principle, right?
Right. And I have to keep reminding students to this day that PhD programs provide a stipend. So that's wonderful. I love those stories. Thank you for sharing them. One of the reasons I was particularly excited to have you here is to talk a little bit about fungi. And the problem that we have is that most students get very, very little of an introduction to it. They know about mushrooms. If they might know a little bit about yeast, they might even know Saccharomyces as its real name. They might even know Schizosaccharomyces. They might even know Eustalagomatus, or Neurospora, but generally speaking, it's not taught very much. And I know that you've spent quite a bit of time in your career looking about fungi. And so what I thought would be good to do is to talk a little bit about what attracted you to that general area of study first. And then, as I think you and I have discussed, a little bit about a positive sort of discovery involving fungi, and then some of the more dire things that we might face in our future.
Absolutely. So how did I end up in fungi? The answer was simple. I went to medical school and I believe in doing everything. So I went and did a residency. And I can tell you that my formative years as a physician was in the height of the AIDS epidemic. And I simply could not believe that we could not do anything from many of people that were at the time younger than I am. And so what happened was that I went into an immunology lab to do my postdoc. And this is the laboratory of Matthew Scharf at Albert Einstein College of Medicine. And Matty was very good to me. I said to him, I want to get involved in something that has direct clinical applications because what's happening in the hospitals is just terrible. And he was running an antibody lab. And we decided to try to make a therapeutic antibody for cryptococcosis. And this is a fungus, Cryptococcus neoformans, that affects people with very severe immunodeficiency. And the antibody was made. And I can tell you that 10 years later, it went to a clinical trial. But it eventually did not get pursued because the problem largely went away in that people who take antiretroviral therapy have their immunity come back. So then the problem of cryptococcosis by the late 90s was not what it was in the mid 1980s. But the experience worked out in many ways because we discovered a lot of basic immunology that was not anticipated. So working in a strictly utilitarian problem, develop a monoclonal for treating patients, opened up a whole set of vistas in immunology that led us to do a work that we wouldn't have done otherwise.
I well remember that period of time, and I lived just south of San Francisco, so I watched exactly what you're discussing. So I myself have never done anything at all, ever, that could possibly improve the human condition. So I tip my hat to you.
I think you're teaching a lot of people about microbiology, and I think you are doing a great job and changing people's lives. And I think that you are improving the human condition.
Well, I'm certainly opening minds. So I tell myself. So here's the issue. When many people think about fungi in general, they think about mold, they think about mushrooms, they think about, and this is part and parcel of my concern about the way microbiology is often in general preceded with the idea of germs or the idea of bad microbes. And I don't care for all of it. And I know that you believe the same way because we've had this discussion. It's all contextual. But you have a fascinating story that it really caught my attention about endothermy in fungi. And I hope you can talk a little bit about that now.
So before we go there, I want to give the credit for this to the first author, Radamés Cordero. And the story begins in lockdown during the pandemic and a thermal camera. You needed two things, lockdown in the pandemic and a thermal camera. Why did we have a thermal camera? Because Radamés was studying the thermal properties of melanin. Melanin is a pigment in our skin that is also made by fungi. It's a remarkable pigment, and he was looking at this with a thermal camera, and he decided to go for a walk in the woods and take with him the camera. And as he was going in the woods, he began shooting pictures. And he was amazed to see how cold the mushrooms were. And he showed me the pictures, and it turns out that had been studied and was known. But it was kind of a fascinating observation, how cooler these mushrooms are relative to the ambient temperature. And he then went on, and I think, to look at the problem in microscopic fungi, that is fungi that you grow in the laboratory in agar plates, and again they're cooler. And it's a great story. It's an entire kingdom, and I be careful when I say an entire kingdom, because the kingdom is huge, and we only sample a few. It's possible that hidden out there are some that are endothermic or very different, but at least among the ones that we looked at, it is remarkable that these representatives of the kingdom work so hard to be cool and to be cooler than the environment. And they use for that a very simple mechanism. It is the mechanism that makes you shiver when you come out of the shower. So even though you may take a bath at a place in which it's steamy and the temperature is hot, you know very well that layer of water on the skin will make you shiver, make you feel cool, and that's evaporation. Evaporation takes out a huge amount of heat. And what the fungi do is they evaporate a lot of water and they keep themselves cool by that mechanism.
That thermal camera work is just amazing because definitely cooler than the environment. Still true on Petri dishes. I would have thought, and I would be wrong, I would have thought that this might have been architectural when you look at, say, the gill structure of mushrooms. But that doesn't explain colonies on plates being cooler.
That's right, Mark. And but what is really interesting is, among the pictures that I sent you, you could see that there are thermal profiles. And to me, the interesting thing is that the edge of the colony is warmer. And what that suggests to me is that something is going on at the edge. And the fungi put out a lot of enzymes. They tend to digest their environment. So we think, although honestly we haven't proven it, that what's going on the edge is heat generated by the chemical reactions of the enzymes put out by the fungus, breaking down the nutrients in the agar.
I have so many ideas about this. I have to rein myself in. This is just amazing. And it's one thing to think about, I hate using the T word of thermodynamics, but it's relevant. I mean, when I teach students in intro biology about uncoupling in mitochondria, say for brown fat, as a way of generating heat, this is not that different in terms of there's going to be waste heat from some reactions, and there are going to be endothermic things that steal heat too. So this is the edge of something really fascinating. I also want to point out this is a great example of serendipitous research, just looking. Admittedly, you had an expensive piece of equipment to look with, but it's still just looking. and I cannot pronounce his name. He's the fellow who got the Nobel Prize for vitamin C. But he said something once about to see what other people see, but think what no one else has thought. And this is where you're at, I think, with this project.
Right.
And
I mean, I think that if you look in the literature, we give credit to the fact that other people had described it by putting thermocouples into mushrooms. What's different is that when you take a thermal camera and you take a picture, you have an image. And you have that image of the cold mushroom against a warmer background. And you know what they say, a picture is 1,000 words. But once you see it, it makes you, and it triggers a lot of ideas. You know, why is this? One of the possibilities that I like to think is that there is something physiologically, biochemically different about this kingdom that prefers its reactions to happen at a colder than the ambient temperature. And from the point of view of medicine, you got to understand that many of these fungal pathogens are coming directly from the environment. So think about the enormous adjustment that a fungus has to make as it goes from the environment at a place in which the fungus may be preferably colder to the temperature of a mammal, 37 degrees. And I think that this is an entire area that could be exploited potentially in drug discovery, because you could imagine looking for the adaptations. And if you can interfere with the adaptations, well, they're not going to do well.
This is an amazing story. I've always been interested with the morphology and structure of bacterial colonies. I haven't spent a lot of time thinking about fungal colonies on plates because it's been many years since I played with yeast, let alone Neurospora. But we tend not to think about the geography of the structure. And this is a fascinating thing that's right at the bridge, if you think about it, of physical chemistry, made me shiver, and an architecture almost. And that's just fascinating to me. But what I wanted to bring up now is that you took this idea with your co-authors and they created something practical out of it. And I love this story.
The microcooler.
Yes.
So we had fun. I mean, science should be fun. And Radamés got himself a styrofoam box and put in a fan from like a computer fan. I forgot where he got it from. And went to the store and got himself mushrooms and put the mushrooms in. And as long as those mushrooms have water, the temperature dropped precipitously. And what do I mean by that? It's like 10 degrees centigrade. I mean, this is a big drop. We're not talking about a couple of degrees centigrade. It suggests the potential of applications. You could imagine a microcooler that allows you to cool certain things, and then when the mushrooms dry up, you just cook them and eat them.
So Arturo, I know that when you blow a fan across a tray of water, you get evaporative cooling. So the question that I think a lot of people are going to have about this is, how is it different with regard to the mushrooms? Are they being a passive series of tubes through which this is happening? Are they expending energy when they're doing it? That kind of thing.
Mark, that's an excellent question. In fact, I believe reviewers have brought up the same issue. The difference is that the gills have tremendous surface area. So when you get a bunch of mushrooms in there, you have a lot of space to evaporate. And this is why the temperature drops so rapidly. Are they pumping out the water themselves? We don't know that, but they may. They may be, and they may be using energy to do so. But because for some reason, being cool is so important to them.
You know, it's interesting further because you mentioned that colonies on plates are also doing cooling, but they don't have gills like a mushroom.
Right. but in that situation, we looked at the fungal colonies. The one thing we haven't done is we haven't looked to see if there are, for example, undulations or things that allow so. That's one possibility. The other possibility is what was mentioned before, that there is an active water pump. That would be very interesting. That is, that these things are just pumping it out. If we had been more observant, and other people have now noticed that already, the story was always there, because when you grow fungal colonies, you often see little droplets forming on the top of the agar cover. And it's interesting because once we published it, a lot of people said, I always saw those colonies. I was wondering what was going on. And there is a picture of that in the paper, and you could just see that they're there. And it's happening right on top of the colony. It's not like the agar is putting out water that is condensing. It's just the water. The colony is putting out essentially so much water relative to everything that is condensing right on top.
That's amazing. I love the idea of a micro refrigerator. That's just the greatest. Now, you did mention something a little bit earlier, and this segues into a second topic, if you don't mind. And it's this, that there appears to be kind of an innate resistance that warm-blooded animals have, generally speaking, to fungal diseases because of temperature. Is that something that you would agree with?
Well, yes. In fact, again, this insight came from very simple research. I went to a meeting in Belgium, I think it was, or the Netherlands, and I ran into a collaborator. His name is Vincent, who's a bioinformatics individual, and we had a conversation. And I presented this idea. At the time, was nothing more than an idea that our temperatures could keep out a lot of the fungal world, and he had access to a collection. And when you put an organism in a collection, they often do a temperature response. And he was able to go into the collection. He wrote the software, and he showed, this was a paper published in the Journal of Infectious Disease, I think in 2009 or 2010, with Vincent as first author, that the majority of fungi do very well at up to 30 degrees. But once you get above 30 degrees, there is a very rapid drop in their ability to grow, such that by the time you get to 37 degrees, you have excluded about 95% of all fungal species. Think about that. Think about the protection that you're getting just walking around at 37 degrees.
That is amazing.
This raises a bunch of fascinating questions. I mean, one of the other fun pieces of research, I call fun pieces of research the type of research I get up in the morning for, but I don't ever have to write a grant because it would never be funded. It would be the idea that why mammals? What are we doing here? As you know, people say, well, after the asteroid hit, a space was created and mammals took over the world. That doesn't make any sense to me. We have iguanas today. How come we didn't have a second reptilian age? And then reading outside my field, I ran into this paper that there was a fungal bloom at the Cretaceous. And this began to put two and two together. And could it be that the mammalian predominance was the result of a fungal filter that kept down the ectotherms and allowed the warmer in? Because, Mark, we don't make a lot of sense. We have to eat all the time. Think about it. If we were ectotherms, if we were reptilian, you could probably get away with one meal a week. So it's something to think about. But that raises the possibility now we go forward. And one of the things that we did, this was done with my collaborator, Aviv Bergman. He's a mathematician, and he took the benefit of being warm versus the cost of being warm. And he asked, what is the optimal temperature by which you don't have to eat all the time, but you still keep out most of the fungi? And that temperature was 37 degrees. So suggesting that there may have been more to this. Now, it could be coincidence, right? But it wasn't 27 and it wasn't 48. It was 37 degrees. So, it's all circumstantial, but it's the kind of thing that I went into science. It's that I like to think about, making kind of this connection. So we may be here because of the fungi. And this also explains why we have such resistance to the fungi, that nobody worries about dying of fungal disease.
I have to tell you that I grew up reading a lot of science fiction. I don't know if you did, but one of the authors that meant a lot to me when I was a boy, Robert Heinlein, had a bunch of little epigrams or quotes. And one of them that stuck with me is this one: specialization is for insects. The idea being that we need to have broad interests. Now, I well understand that it's important to be able to specialize and to really understand things deeply. I'm not disrespecting that. What I am saying is that historically, real advances in science have come from bringing different groups of people together with different outlooks. The very founding of molecular biology was set up by quantum physicists. Really was. People like Hans Delbrück and people of that nature. So I would argue that being willing to look outside the box, as it were, outside the petri dish, whatever your metaphor would be, is really critical. And this is another great example. I don't know if there's ample evidence for this fungal bloom at post-Cretaceous, but I do know that climate changed all over the planet all at once. And so presumably there's some kind of evidence for that taking place, and I wouldn't be at all surprised.
Well, the fossil evidence shows that there was a fungal bloom. In fact,
that's fabulous.
Whenever the Earth does poorly, the fungi do well. There is a 1 meter of spores at a mountain in South Africa after the Permian extinction.
A Permian?
A Permian extinction. You still
Every time?
You still can see the fungal record. Fungi do well when everybody else does poorly, at least in the geologic record.
I won't even talk about those enormous fungi that used to exist too, that were 20, 30 feet tall.
The land.
Yeah. But this is fascinating. And the reason that this might be important is because as we get changes in our climate, it makes me wonder, because I like to say that the hand of Darwin is on all that lives. And as the temperature rises, is that not an opportunity to fungi to adapt themselves to slightly higher temperatures, which might in turn give them a better ability to cause us problems?
Absolutely. I mean, think about it. We are protected against the fungi by 2 pillars. One of them is temperature, and the other one is advanced immunity. But frogs have advanced immunity, and they've been decimated by a chytrid fungus. But you can take those frogs that infect them and bring them into the lab and put them at 37 degrees, and it cures them. Temperature and high immunity gives us tremendous protection against the fungi. But so we learned in the late 20th century that if you became immunosuppressed, fungal diseases went up. You weaken one of the 2 pillars: AIDS, transplants, immunosuppressive drugs. But what would happen if the fungal world adapts such that organisms that today are not pathogenic because they can, for example, grow past 35 degrees centigrade, are able to adapt a few degrees because of the temperature? Then we may be running into whole new classes of fungal pathogens that we can't even imagine.
Again, this is something interesting, and you might not think it's relevant, but my mind goes everywhere too, so I think we're in good company.
Good.
One of the early science fiction novels that Robert Heinlein wrote, and it was at a time when people didn't know much about Venus. The idea was Venus being very hot and humid, and the major pathogens that killed people in days were fungi. And that's kind of amusing.
It's interesting because sometimes when I give talks to students, I give them a picture of Venus and a picture of Mars. And I said, say that life exists in both. Which one would be potentially more deadly to you? And they always picked Mars. But Mars is pretty cold. You're likely that if those things get into your blood, you're likely to boil them. But imagine that there is life in the clouds of Venus. All you have to do is take microbes and put on them some sort of vacuole so that they float in that atmosphere. Those are temperature adapted, though your temperature may not protect you against an assault from Venus.
And what's interesting to me is so many fungi have very non-specific ways that they attack. So it's not the key in the lock that we're so used to with a lot of, say, viral
Right.
or bacterial disorders.
Absolutely. The fungi degrade their environment. They are the big degraders. They are the things that break down a lot of biomass. How do they do that? They put out a lot of enzymes that break down, all the constituents of plants, the constituents of animals. So when they get a hold on people, they digest a lot of the tissue, and that makes them very hard to get rid of. Because if your
Amazing.
tissues is dying, how do you even get antifungals in there?
Now this gets me thinking about, I know you've worked with a couple of arthropod systems to study fungal diseases. And now it makes me wonder if anyone is starting to try and adapt fungi to higher temperatures by directed evolution in the lab.
Well, I know that there's a report from a company who actually successfully did this because the arthropods are smart. They sun themselves. Think about it. They find a hot rock and they can give themselves a fever. So, and this paper showed that in fact you could do something like this in a matter of months. That you could do adaptive evolution. But we have a preprint that was deposited last month from a very, very bright postdoctoral fellow in my lab, Daniel Smith, in which he walked around Baltimore and he collected fungi from warmer and cooler neighborhoods. And Mark, they are already adapting.
Yeah.
The fungi from the warmer neighborhoods are more heat tolerant. And what that's telling you, even within a city, which are, cities are heat islands because they get things are already happening. So I think that we need to keep an eye on what's going on out there. Otherwise, we may be very unpleasantly surprised.
I love the story that you told about finding more thermotolerant fungi on the warmer side of Baltimore. That sounds like the beginning of a joke, and it's still true. There are many locations on the planet, I think, that are above 37 degrees C all the time. How about those fungi? What's going on with them so they're able to survive and perhaps create a different kind of situation there? And in addition, are they then able to perhaps, although this might require a suite of genes rather than just a few, could they use the equivalent of horizontal gene transfer to move that to non-thermotolerant fungi?
So let me answer your second question first. I don't know. I agree with you that probably thermal adaptation is a lot of tweaks in different places. So it's hard for me to see, one gene being transferred. But the best answer is I don't know. But let's get to the first thing. Most pathogenic fungi are near the equator. So, of course, there are many fungi that can grow at temperatures higher than 37. In fact, the aspergillas grow in your compost, and that's why the compost gets so hot. But it's an statistical issue. The majority of fungi cannot grow at 37. And the ones that can grow, for them to be able to cause disease, would have to have mechanisms to undermine the immune system, to be able to survive in the host. So you need two
legs,
things together. And for most of the fungi, they are environmental organisms. They don't necessarily gain very much. There is a lot of selection pressure for them to be pathogenic for anything.
I agree with that. I think it's an interesting topic. And what I like best about what you're discussing is that for each of the topics we've discussed so far, there are some very accessible papers that I can put in the show notes that interested people can read about. So I'm really appreciative about this. Do you have any exciting things that are in the cooker right now, research-wise?
Well, I find everything exciting. So it's hard for me to discriminate. We have some very nice studies on the structure of the capsule polysaccharide. We have other things percolating, but they tend to be more specialized, right? We're talking about Baltimore. We're talking about the Permian. We're talking about the Cretaceous. So to me, I'm interested in the whole range of things.
Yes. I think it's amazing. And the problem is that specialization is one that's been so encouraged. And again, it's very easy for that to sound like I'm criticizing, and I'm not in charge of anything. But I do know that enthusiasm is what really drives science.
That's right, Mark. And I think there is a good reason for specialization, and that is that things are so complex that in order to get deeply, you have to really become very specialized. The problem is that we are teaching and fostering specialization without a concomitant effort to encourage the generalist in every scientist. That, to me, is the problem. I don't want to replace what we're doing. I want to expand it. And people should have some degree of license to wonder and to think about other things. And what happens is scientists are often very uncomfortable with people who do multiple things. Well, they're not serious, right? Because what are they doing? And in fact, scientists who have contributed in multiple areas often are not as well known or regarded as individuals who basically worked on a problem for a very long time. I don't know why that is. I think one possibility is that science is so hard that people have trouble with the idea that anybody could really contribute in multiple areas. But I will tell you that I went into science because I was interested in many questions. And even though the system tells you focus, 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 I listen, I focus. My grants are very focused. I refuse to let the breadth of knowledge not engage. And I try, the advice I give to everyone is every day, read something outside your field. 5 minutes. If you do that for a year, you have 365 new facts. And after a while, your brain will begin to make connections. And it's a wonderful thing to do, to be able to see the grandeur of science, the grandeur of biology in ways, geology, things that one not necessarily is what we do each day.
Well, they're all connected.
Absolutely.
And why wouldn't they be? May I tell you something that happened to me when I was in middle school, and it was transformative?
Please.
We called it junior high school. And our biology teacher or our science teacher wanted us all to do projects. And I was like, a science fiction nerd. And I'd heard this story that you could take flatworms, you could train them to run a maze, you could cut the trained one up into little pieces, feed an untrained worm, and then that worm would know all that stuff. Now, of course, I learned many things trying this. One is it's really hard to build a maze for flatworms, right? It's even harder to train them. But then I thought, what did I have in terms of the materials that I had? And this is before I read journal articles, and my science teacher certainly knew nothing about it. And I discovered these flatworms, these planarians, like to live on the underside of rocks. So I taught them, and I'm sure this would not be approved today, to associate darkness with an electrical shock. So if you divided a petri dish in two, they would collect on the lit side. And I showed they could remember this for long periods of time. And I couldn't do the feeding experiment. It never worked out right. But I thought, I've got to get something. And I remembered that planarians have the ability to regenerate. So they have a simple head with eyes and a primitive brain. And they have not a notochord, but they have these two neural lines running down. I cut them in two. The head half regenerated a tail. The tail half regenerated a head. But both of them remembered. And I didn't know this was a big deal. There was no reason I knew this was a big deal. And I did this when I was, 13? And what happened is I won a big award for it. My parents thought it was stupid. Why do I have all that smelly stuff in my room? That's just the minor part of the smelly stuff, if you know 13-year-olds. But what was interesting is I got a letter from Roger Sperry at Caltech, who said, "You think like a scientist."
He was a Nobel Prize winner.
That certainly was in neuroscience. And though I don't do work in that area, the point I'm trying to make is many people think that science is about complicated machinery, and it can be. But it's also about a way of thinking and a way of designing and a way of interpreting. In some ways, we're all scientists. And that's what I try and teach students to think of. So, for example, when my students in microbiology write a term paper, I call it a microbiography because that's me. I have them do a one-page version of it for their English major pals. And between you and I, Arturo, it's a trick because when they talk at that level of what they're learning, number one, they understand it better. Number two, they learn new things from that person outside the field every time. So this idea of science communication is so critical, and that's something you've done a lot of. See how I segued that?
You have done it too. I mean, I think it's really, look, we scientists are embedded in a society that supports science. And it's really, really important that we communicate and that we communicate in ways that resonate with people. And it doesn't come naturally. We are trained to become very specialized very quickly. I often joke, you want to ruin a good dinner party, ask a scientist to come in and tell you about their work. After the second sentence, they may get very specialized. Now, that's an exaggeration. There are many scientists who can get up there and enthrall a dinner party. But it's just an example that we need to, in many ways, part of training should be how to get ideas across. Mm-hmm. And that's really important, and it's underdeveloped in most programs.
So a really good example of this, because I get to teach microbiology every year, and I have a small class, and it's very discussion based, and we have a lot of fun. That's why I call them micronauts. That's just fun for me. But I use a lot of these short essays that you have written in my classes. And a good example is that, and I believe that we can show an image of this, you wrote a very nice article about the term pathogen in Nature. And what was interesting is I always assign that paper. I have students read about it and respond to it. And what was interesting is that one of my students, in writing about it, actually misspelled pathogen and called it pathogon, which is exactly right. And in fact, you responded to a couple of tweets on that, as you can see here. My point is, we need to have this kind of ability to put concepts across to people. They're not too complicated for my students to understand. This idea of understanding the definition of pathogen is one of relationships, not a unitary thing. So as you've heard me say before, to a real estate agent, it's location, location, location. And to an organism that causes disease, it's context, context, context. And this is the value of what you've done. And I
mean, thank you, Mark, but I also will tell you that you're one of the very few people who got it, who got it right away. Look, it's very hard for the way that we're trained to think we try to assign causes. And what happens is that we try also to divide things. These are pathogen, non-pathogens, and these are whatever. And we need categorization. And in doing so, we begin to make mistake after mistake. The word pathogen is a problem because it gives a microbe a trait that is not its own. No microbe can be a pathogen without a host. It needs a susceptible host, period. I'm old enough to be vaccinated against smallpox. It doesn't matter. Smallpox is not a pathogen for me. It's just an example. And what happens is that if we think using words, I mean, I certainly can't think without words. And if there is a problem with the words, there is the potential that there is a problem with the thinking. And what are we worried about in that article is that unless we have these words right, that people may be designing experiments, trying to make vaccines, etc., with flawed concepts, and that may inhibit progress. By the way, I also want to give Lysiane Porofsky, she was my co-author on that. And for almost 30 years, we've been collaborating, and she gets equal billing for all of this.
Well, I think it's wonderful that you've done, I can't, 10, 15 of these short essays of like 2 pages. There was one that I assigned to my class about living inside another cell and what the invader, for lack of a better term, has to perceive to deal with that, the adaptations that they need to make. And it's a very transactional view, and it's an important one, which brings me to this. You recently wrote a book with Dr. Fang, and here is an image of it. And I am going to get you to sign my copy. It's going to happen.
Absolutely. Next time I see you, at ASM or something, as the 2 escalators pass, you say,
That's right.
Arturo, come up or wait for me down there, and I'll go down and sign it.
You sign at the top and throw it back down. Right, exactly.
Yeah, Ferric is a wonderful colleague and collaborator. We got to know each other when we were both editors of Infection Immunity, and we began to write about the problems in science. The first paper we wrote was because I was fed up of people dismissing work by calling things descriptive. Well, that's descriptive science. It shouldn't be published. What are you talking about? Why don't we throw out all of astronomy? All of the sciences that are, throw out all of paleontology, And what came to realization was that people didn't even understand the words. So for some, description meant that they weren't doing cause and effect. For others, description meant that they weren't doing going deeply. But then we collaborated for almost 20 years writing essays. And what happened in the past year was that we decided to get them all together, update them. And there are 30 plus essays there dealing from everything from COVID to what is science. And it's been one of the most rewarding things I've done in my life, to sit down and write. And I learn a lot, and we're already working on the 2nd edition.
Oh, well, I've got to tell you, it's been so useful to me. And I've really appreciated, as we start to wind down our time today, I normally ask visitors what one thing sold them on microbiology, but you've already answered that question. And I found that to be very, very interesting. And like I said, very selfless, because I remember that period very well. And believe me, I thought it was awful, but I thought it was going to be a lot worse for quite a while. And who knows
if there had been no AIDS, I probably would have gone into cancer.
Yeah.
I remember, what are the big problems? What are the big degenerative diseases, Alzheimer's and things like that. And then I went into the wars, and I just could not believe that a disease had come out of nowhere and was killing people who a few months earlier were totally healthy. Yes. And we couldn't do anything about it. And so that was a really formative. And Mark, the other thing that was very formative and gave me tremendous optimism was that I not only watched that, but I watched the result of medical research that within 6 or 7 years generated the first antiviral. And by the 1990s, it generated multiple antivirals. And this disease that had been uniformly fatal became very manageable. And today, people that take their medicines with HIV have the same life expectancy. And I think about that because that gives me a tremendous amount of hope. And it shows you what humanity can do with science.
Absolutely. We tend to, some of us have a love affair with the negative. And it's very easy to think that way, to look that way. And social media helps us with that negativity, I think. But at the same time, there's a lot of positivity, a lot of successes everywhere if you take the time to look. I wish I had the actual expression, but sometimes bad news shouts and good news whispers. So we need to change our ears a little bit.
I just leave you. I find that science is the best antidote. We actually use this expression in the book, the best antidote for depression. There is so much that you can celebrate what the human mind can do and has done. And I'll just give you a little anecdote. February 16th, 2016. Around there, they first announced gravitational waves. Think about it. This happened a billion years ago. Life in this planet was unicellular. During that, these waves travel through space and time for a billion years. And during this time, a species emerges that learns about science and builds an interferometer. And then the universe is aware of itself. And that is a tremendous lift as I read the paper and I see bad news after bad news. And I think of what we have done. And it gives me a lot of hope and a lot of optimism for the future.
No, I agree completely. You know, in even in the small microscopic, oh, that's a pun, microscopic area, I was listening to This Week in Microbiology recently, and one of the people, I think it was Petra, was saying that there was such a positive impact of students when they discover something, when they observe something, not a matter of like checking off a box or doing an exercise, but something where no one knows the answer and they determine it. I've been saying this for many, many years. It's transformative to students no matter what it is that they want to do or study or become. I mean, there's such enthusiasm that you feel when you see it, and it's contagious. And I think we all need to take a moment and listen for that.
Absolutely, Mark. And you do it beautifully.
Well, Arturo, it's been just a delight to have you here. I really appreciate your time. I'm a big admirer, and I want to wish you the very best, and I hope we can talk again soon.
Absolutely. There is a lot to talk about. Maybe in the future you'll have me back. Thank you. I had a wonderful conversation and really appreciate that you had me in your program.
Thanks so much, and my best to you and your family.
Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, comments, or suggestions to me at mattersmicrobial@gmail.com. Show notes with so many tasty links can be found as usual at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Arturo Casadevall is chair of molecular microbiology and immunology at Johns Hopkins University in Baltimore, Maryland. Many, many thanks to David Renata for superb editing, and to Reber Clark, as always, for his wonderfully quirky music. I hope you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.